All right, everybody, we'll get started today. So welcome to the Spring 2023 GES Symposium. Today we have 7 student speakers. They'll all be presenting the results of their research experience. The format will go as follows. So first, the student's mentor will come up and say a few words about the experience for the student. Then the student will give their presentation, sharing their results. After that, we'll take questions from the audience, and I'll be moderating the questions. The talks are about 10 to 12 minutes long. Once we're done with all the presentation, we will move outside to the lanai, and everybody's welcome to join us for food and drink to celebrate with our presenters. Some other housekeeping stuff. If you do need to leave, please try to time that for in between speakers and not during their talk. And also at this time, please take a moment to silence your cell phones if you haven't done so already. That way we don't have interruptions while the speakers are talking. Finally, I'd like to acknowledge the support of our large ohana that helps pull all this off. Lentina, who's in the back, our student service specialist, keeps you in line. All of our ocean office staff, so Kristen, Maria, Batalpa, Roberta, Bryce, and Han. And then obviously the ocean faculty who help guide the overall program, and all the mentors, and of course family and friends that are critical in the support of these students when they're doing their research experience. So without further ado, I'll have Dr. Toonen come up and introduce Jasmine.
All right, so today it is my great pleasure to introduce Jasmine Awaya. So Jasmine is a local Kailua girl, grew up in the ocean, spending her time always passionate about coral reefs and marine biology. Always wanted to be a marine biologist when she grew up. And usually when I do GES students, I sort of figure out a project in advance, do the program and recruit someone to do that. And Jasmine just emailed me out of the blue. I had decided I was way too busy this year. I wasn't going to do it. And she's so awesome that she convinced me to do it anyway. She started a project. She has been working out at Mo'oloi, trying to understand how to better help coral reef restoration. Please join me in welcoming Jazz.
Okay, aloha everyone. My name is Jasmine Awaya, and today I'm going to talk to you about my project looking at fragment size and density and the effects on the growth and survivorship of the coral species Montipora capitata. And I was fortunate enough to be advised by Dr. Toonen. So a little bit of overview of how this presentation is going to go. We're going to touch on a little bit of importance on corals, as well as history of corals in Kaneohe Bay, as well as the methods being focused on, the where, the how, and the what of this project, results, what do these results mean, and what is our suggestion moving forward in the future. So the big question is often why? Why is this important? Ultimately, it's for restoration efforts. Corals play a large, crucial role in the marine ecosystem, providing about 25% of the biodiversity in the ocean. And all these species that rely so heavily on this ecosystem are often food sources for us, as well as corals have a large role in having a structure on the sea floor, which impact wave formation, ultimately impact our marine or coastal regions. However, global health of these coral reefs are in massive decline, and as we've seen, a 50% loss globally. And because of its ecological importance, as well as its massive degradation, we are seeking active restoration efforts to help save these ecosystems. So microfragmentation is the method of restoration that I focused on in this project. It is one of the most widely used methods of asexual reproduction or replication, and it involves cutting a colony of coral into smaller pieces, which the goal in turn is to be outplanted or cultured in the natural environment. And the reason why this process works is because corals grow from their live edge. So when you're cutting a colony of coral into smaller pieces, you're increasing that surface area of potential growth. So in this image above, in this image above, there is 4 individual fragments, and throughout time, you can see they grow and they fuse together to become one colony again. So a little history of corals in Kaneohe Bay. Throughout history, they've experienced a major amount of anthropogenic and natural disturbances. A few examples include dredging. As you can see in your lower right-hand corner, there is a black and white, there's a black and white aerial view of Kaneohe Bay. And in these circled areas, I know it's a little bit difficult to see, but these are where areas where dredging had occurred. There has been sewage discharge events from 1930 to 1975, as well as consecutive bleaching events that occurred in 2014 and 2015. In this upper image above, this is 2 images of the same patch of reef, one taken in October and the other taken in December. As you can see, just based off color alone, how much resilience that these corals presented. Just in 2 months, they were able to recover quite fast. And because of this interaction, this behavior that is noticed, Kaneohe Bay has been a place of extensive research to see why corals behave this way. 2 main reef building species in Kaneohe Bay are Varieties compressa, as well as Montipora capitata. And so we go on to say the species that I focused on was Montipora capitata. It's also known as rice coral, and it's an important reef builder. It is found in the Indo-Pacific, but due to its preference in shallow, warm, and turbulent waters, it is more common in Hawaii, and compared to other places in the Pacific, it's harder to find. This species comes in 3 different growth forms. It comes in our applauding growth form, our branching growth form, and our encrusting growth form. So a little bit about methods and what we did to do this setup of this project. This entire experiment was conducted on the east side of Oahu in Kaneohe Bay at the Hawaii Institute of Marine Biology. The island itself, the Hawaiian name for this island is Moku'oloe, and it is commonly known as Coconut Island. The entirety of this experiment, from assembly to monitoring to analysis, was all conducted up at the coral nursery, denoted in the red circle above. So a little bit about our setup. We used a diamond bandsaw to cut a single colony of Montipora capitata into 3 different size classes, being small, medium, and large, which correlate to our 1, 4, and 9 centimeter square fragments. We then took these fragments and glued them onto a terracotta tile in the specific orientation, depending on if we were looking at a low or high density treatment. And each treatment experienced a total of 10 replicates, giving us a total of 60 tiles and a grand total of 220 fragments. We then took these fragments, these tiles, and placed them on a floating PVC pipe rack in the coral nursery as such. So a little bit about our data collection. This was an 8-month-long process. We gave these corals an acclimatization period of 2 weeks, where we let them rest, recover, and respond to the initial stress that was placed on them during assembly. And then after that, we started taking data for growth as well as survivorship. So for our growth data, we collected this data taking aerial photographs in the corner about once a month. In these photographs, we included the tile itself, a ruler, as well as a color bar that would be used for analysis to ensure that each photo had the same distance and angle from the tile. We fixated the camera onto a mount, as well as being these photos were taken inside the coral nursery. It created a softer lighting, not only for the health of these corals to prevent harsh lighting and heat, but also these photos would be easier to analyze down the road. For survivorship, we collected this data in the water 1 to 2 times a week through visual observations. If there was any live tissue on the fragments, we would still consider them alive. However, if it was complete skeleton, we would denote them as dead. And then we shuffled these tiles. Once we collected this data, we shuffled all the tiles on the rack to ensure that each treatment had variable and I would say fair conditions in the natural environment. So a little bit about our data analysis for growth. We used those monthly photographs that we took and we placed them into ImageJ. We then measured the total surface area of live tissue that was on each fragment on every tile. So as you can see, this is an example of how we use ImageJ. This line, we click all the way around each fragment to show, and then we then measure this. So you can imagine when you're doing this for 220 fragments, how long this takes. But after we did that, we summed up the total live area for each tile, and then we ended up comparing the final to initial surface area to get our percent change in growth. For survivorship, we use those weekly observations to calculate the percent survivorship, and we took both of these data sets into RStudio and ran a ANOVAs and pairwise comparison to get our results. So for our results, we were able to get these plots here. On these plots, we're looking at size class. So we're looking at our small, medium, and large fragments on the x-axis, and then our percent survivorship as well as our percent change in growth on the y-axis. A little bit of background for box and whisker plots for those of you who are unfamiliar. This is our median value, so that means 50% of our data is below and above. This is our upper quartile and our lower quartile, as well as our max and min values, and values such as like this are our outliers. And so when we're looking at these plots here, you can say that size class had a significant impact on survivorship. So given with a p-value of 0.01, with larger fragments having a greater percent survivorship compared to smaller fragments. However, when we look at size class versus percent change in growth, we do not see a significant impact as growth rates, the median growth rates are fairly similar to one another. And we can statistically back this up with a p-value of 0.59. For density, we are looking at our low and high density treatment. And similarly with the previous slide, we're looking at percent survivorship as well as percent change in growth on the y-axis. For density, we did not see a significant impact on neither the percent survivorship nor the percent change in growth. And we can statistically state it with our p-value of 0.48 as well as 0.1. So for our discussion, we're kind of summarizing this data. What do these plots mean? So with size class, we can say larger fragments have an increase in survivorship, while smaller fragments have a decrease in survivorship. And this could be due to initial stress with a greater proportion of damage. As you can see on this image on the side, this is the size of a larger fragment, and with just a small corner of damage, this could be an entirety of a small fragment. So there's just that greater damage that smaller fragments face, as well as predation. These smaller fragments are perfect bite size for parrotfish to come around and pick these tiles, these fragments. So they are just more prone to predation as well. For growth, there was no significant impact in terms of size class, as growth rates were fairly similar, like previously mentioned. And then there's this idea of trade-off between new growth and maintaining the existing tissue that is already on these fragments. For density, there was no significant impact. However, we did experience slight trends in our data. We could see that there was a slight increase in survivorship with the high density treatment, as well as a slight increase in growth in our low density treatment. And the idea with density is that you kind of wanted to observe that fusion, that interaction between between the fragments. However, with our experiment, we did not get to see that interaction. However, maybe if we conducted this experiment for a little bit longer, we might have experienced that. So with all that being said and done, our recommendation would be a medium-sized fragment with a low density treatment would be the most optimal restoration startup conditions for the species Montipora and capitata. And these are just two examples of a single tile from the beginning to the end of how these go through. And with that being said, I would like to acknowledge my advisor, Dr. Robert Toonen, as well as Chris Sikowski, Claire Lewis, and Claire Bardine for helping me with analyzing, data collection, and writing up my thesis, as well as my parents, Nana and Tim Awaya, for being here and sticking with me through this amazing and wild journey of an undergraduate degree. Thank you. Any questions?
Questions?
Yeah.
Great song. I'm just curious, how would you apply the restoration techniques from, like, so the Canada Bay is a pretty unique ecosystem, and I'm assuming that the corals that you study at Canada Bay, how do you think other ecosystems around Oahu might differ in the coral restoration?
Right. So with Montipora capitata specifically, places such as like North Shore, they're not going to have that just because it's the waves, the different wave interactions. They're not going to have these species. So places around the island might differ in what species are more prevalent, but specifically in Kaneohe Bay with that Montipora capitata, you can kind of apply it with the species, but maybe you can do other similar type projects with different species that are more prevalent in other ecosystems around the island. Yeah.
You kind of alluded to this at the end, so you showed us the effects of size and the effects of density. Did you see an interaction between those?
Yeah, so we tested that and there was actually no significant when it comes to the combined effects of those interactions. And so we were able to we did look at that and we were able to see that there were in fact separate factors. Any other questions?
Question?
So there's this issue of survivorship. You have a lot of variability in survivorship.
Yes,
Also a lot of variability in how much they do.
right.
And so, science is great, but there's also just watching. And I assume that we're trying to figure out these type of fragmentation approaches, what's going to work. There's some element of like just intuition of what looks good, and like your next step is to follow this curve. So based on just kind of looking at them, I assume this, assessment that you have at the end where you're like, oh, I have medium density or medium size fragments and low density. And is there an element of intuition that makes that decision like this? Look very different.
Well, also, yeah, so one of those things too is, okay, medium-sized fragments, you're kind of maximizing your survivorship with still reducing, minimizing the amount of startup material that you need, especially with the low density treatment, because density we see didn't really play a factor in, we can say, okay, let's use those low density treatments because they didn't really have a significant impact. So we have less amount of startup material that we need for live healthy corals to start up with. So that was a big reason why we chose the medium size, because also reduced area startup. They both had median values, similar medium and large fragments had both similar median survivorship values. So we can kind of maximize that while minimizing startup material. Yeah.
Questions? Great, thank you, Jasmine. Well done.
Thank you.
So I'll have Dr. Dulai come up and introduce our next speaker.
Hello everyone, it is my great pleasure to introduce Brandon Dela Cruz, who will be presenting to you on his work that had its ups and downs. And I first met Brandon when we kind of pitched the project idea to, in general, any student. We welcome the input from anyone, because we wanted a group of students to work in a community setting, to collaborate with them, learn how to work with the community, how to feed the data, and how they can use science to help them out. And while Brandon was working on another project already, he eagerly joined us, and how lucky we were, because he really was the kind of pillar willing to go every single time to sample, collect samples for others and help out. And although he was interested in hydrology, and the community partner needed information on E. coli, so he said, okay, I can work on it. Let's see where that leads us. And as I said, there were some ups and downs, both with data analysis, also navigating what is the appropriate way to present the data that's agreeable to the community partner as well. But here we are, and I'm so proud to present Brendan.
Hello, my name is Brandon. My mentor is Dr. Henrietta Dulai, and my project was doing a spatial temporal analysis of the distribution patterns of E. coli in an urban wetland. Oh, so kind of what I'm going to be talking about is like, what was the driving force behind the project? What were the goals of the research? Why is this location so important? And how is this project executed? What does the data tell us? And kind of some take-home messages. So the motivation behind the project was to address the community partners' concern that they had with E. coli, and kind of to determine like the distribution patterns of E. coli in relation to spatial proximity and urban development, and the temporal differences correlated to environmental and water quality parameters. So the objective was to use the Sea Grant Kulana No'i communication best practices, where we respect our community partners' right to privacy for their data and present it in a form that they see fit. So we did this by doing a multi-step analysis where we characterized the spatial temporal patterns of E. coli with respect to the urban surroundings and variations associated with rain events and water quality parameters. So this is our study site. As you can see, it is an urban wetland. There's a lot of like urbanization occurring all throughout the entire study area. Each point represents the sampling locations where we collected water samples once a month for 11 months. The purple circles represent water parcels. The blue dots represent locations with groundwater discharge, and the orange dots are outflowing catchments. I decided to break the study area up into 3 sectors. So the western sector, central sector, and eastern sector. And this was because it was meant to serve as like a reference. So data collection happened once a month for 11 months. What we did is we used the equipment here, which is in order to collect water samples without contaminating the samples. Additionally, what we did is we used the YSI multi-parameter SON to measure water temperature and specific conductance at each sampling location. We also grabbed precipitation data from the Hawaii Climate Data Portal. So the technical approach behind the analysis was to use the EPA-approved standard method of examining wastewater and water examination. We did the IDEX color test. Basically, what we did is we collected 100 milliliters of sample water. We mixed in the color reagent, dissolved it, poured it into a quantitray, sealed it, incubated it for 24 hours. And the following day, what we were looking at was color change. So the color change will tell us the total coliforms present in each sample. And when you use a UV light and shine it, the wells that change color and fluoresce will tell you the E. coli concentration within each water sample. So after I got all of my data, I did the Z-score standard method, which is a way of normalizing the data and creating a normal distribution for the data that I collected. And this was done by getting the observed value, putting it on a log 10 scale, subtracting it with the mean and dividing it by the standard deviation. And when you get all of that data and you put it on a plot, you get this. It doesn't really tell you that much about like what that all means. So therefore we had to use like other software such as QGIS and Python in order to further analyze the data. So QGIS is a GIS software, which is Geospatial Information System software. And what I did was get the Z-score E. coli data, use the IDW, which is the inverse distance weighted interpolation, and created a representative surface of the densities of E. coli throughout the study area. Additionally, I used Python to conduct the correlation analysis between the Z-score E. coli concentrations and the environmental and water quality parameters. So this is the graph of the daily precipitation during the entire sampling period. So from January all the way to November. So the blue line represents the precipitation that occurred, and the orange dots are the days that we went to go sampling. So the precipitation is put on a log 10 scale to better visualize how much precipitation occurred. And what we can see here is, 10 to the 0 is about 1, and this is in millimeters. So therefore, there was no significant rain events that occurred during the sampling period. So when I did the correlation between the Z-score E. coli and the log 10 of precipitation and created a polynomial fit line, I was able to see that there was no true correlation between precipitation and E. coli. Precipitation can also act as a dilution for the amount of water to dilute the E. coli concentrations. But one thing about precipitation is because of the highly urbanized area around the study area, urban runoff can act as a transport mechanism for going into the study area and causing some E. coli fluxes. Similarly, I did that with the E. coli and temperature. And what we can see is a statistically significant correlation between E. coli and temperature. So with high temperature, that can cause stagnant water bodies, which limits the flushing effect of water. So therefore, causing the E. coli to persist in these environments. Moreover, I also did an analysis with specific conductance. And as we can see, there's very low correlation between specific conductance and the E. coli concentration. This was actually kind of a good thing because since the farm is entirely fed by groundwater discharge, we were able to rule out the springs as a source of E. coli into the urban wetland. So with the data, I also created a heat map. And I thought it would be cool to do a comparison between the wet season and the dry season. So from November to March and April to October, doing the IDW method. And as we can see, that there are some consistencies in where E. coli tends to persist, specifically in the eastern sector of the study area. So Due to the low amount of rain that occurred during the sampling period, it's difficult to determine if there was any true correlation between precipitation and E. coli. Further, there was also no significant relationships between specific conductance and E. coli. However, we can infer that temperature is the best predictor of E. coli concentrations of the 3 correlated parameters. So some of the limitations that we faced during the project was scheduling sampling between our community partners and our research team. Having a cost-effective method to process and analyze water samples. Because of the IDEX kit, we weren't able to determine any strains of E. coli that were present in these samples. And there was no high rain events that occurred, so it's really hard to determine if precipitation really is correlated with E. coli. So some future research direction for this could be to increase the sampling days and maybe have more event-triggered sampling, such as when there's high rain events, we can go out and take some samples and take a look if there's any correlation between precipitation and E. coli. To also maybe use different methods of characterization, such as qPCR, which can help us determine like strains present of E. coli. Just because not all E. coli strains are pathogenic. References. I'd like to thank my mentor, Dr. Henrietta Dulai, Professor Mackenzie Manning, and our external reviewer, Dr. Craig Nelson. Additionally, just the huge support system that I had here at UH Manoa with a lot of the faculty here, my peers, and just my research team that helped me go out and collect water samples. And yeah, so the work was also supported by the National Science Foundation and the Hawaii Pacific Foundation and by Europe. Questions? Yes.
Organizing meetings. There be some scientists where they just go out and collect the activities.
That's a little difficult just because the urban wetland also is a place of agriculture. So a lot of times they're using fertilizers. So we don't want to go out and collect samples when they're distributing nutrients to their plants. Yes.
Do you have any hypotheses for why there was the accumulation of mixture into the pond?
I guess my hypothesis would be because of just the general shape of the study area. So I think I can speculate that the reason why it's like a higher and consistent accumulation here in the eastern sector is because there's a disconnect between this area and the outflowing catchments, which are about here and here. So because of this like shape right here, it kind of prevents and has like no true connection to it. So it kind of like increases like the residence time of E. coli in that area.
Thank you.
Yeah.
Yeah.
Yes.
Hey, Brandon, so,
Yeah, what's up, Jim?
you got a prior picture of the district groundwater discharge study. And wondering if you have a sense, I mean, I know you didn't get any significant rain events, but sense from the topography of the land, if the rain runoff points will be in the same location as the groundwater.
Yeah, just because the layout of the urbanized area, there's like kind of a, I'd estimate probably a wall that's about this big right behind the entire structure. and there's a restaurant, there's parking lots. So that can be kind of where it enters in.
I'm just wondering, like, let's say you have this massive rain
Yeah.
and water runs off into the area.
Mhm.
Would those be the discrete locations, or is it expected just to be running off that area?
That's a good question. I think based on like field observations from what I saw, because the upper wall over here is kind of tall, from what I did see, there are like weep holes within the wall. So I'd assume that that's like kind of the entrance point of where the runoff water could be entering the study area.
Thanks.
Yeah. Any more questions? Yes.
You know, if E. coli is predominantly entering the system, or if it is predominantly growing in the system, like where it's actually coming from and being.
So the thing is, E. coli is actually a fecal indicator bacteria. So it's very ubiquitous throughout tropical climates and exists also like in warm-blooded mammals such as ourselves. It helps in the digestive processes. So it is very common to see E. coli everywhere, including soil. So in this case, it could be not through like contamination, but just from like it being existing. That answers your question. Yes, Quinn.
Hi, Bradley.
Hi, thank you.
Okay, I just want to commend you for working with the communities. Communities are really hard, stepping up and using technology as practices. But have you, or do you have an idea, or have you told them already, any ways they could negate their E. coli concentrations? Especially that top corner.
Yeah, so I did have a meeting with our community partners with Dr. Dulai. We had a discussion about the data and what it actually means for their farm. It's difficult to really like prescribe any like recommendation for them to do. One thing that they potentially could do is maybe like increase the water there, like fresh water, because they do have a sprinkler system in their property. But the only like disadvantage with that is like you're tapping into municipal water supplies, so that can be kind of costly. Yeah.
Great.
Yes.
Well, I just one question, since we only looked at normalized data,
Mhm.
are these numbers high enough to be considered a problem? Or the actual concentrations?
So the actual values of the E. coli is not necessarily, that's a good question. So I mean, the values are material, but I think the raw values, such as what it is not necessarily the most important component of the project. It was kind of just understanding like what was the distribution of E. coli that is occurring in the study area.
Yeah.
Sorry, it was
So the area of dry season.
No, so actually the springs were pretty much relatively zero throughout the entire sampling period, which is good, you know, that's not really like a source of contamination. As we know, you know, groundwater can be kind of hard to tap into and like kind of purify. So it was good to know that the groundwater discharge was not a source of the E. coli. I saw questions. Yeah.
So we found that there was a correlation between E. coli buds and temperature. So my question is this temperature of the water goes to the temperature of the air temperature, or is there something to recommend for the management of this place that change cool down waters?
That's a good question. I mean, also the springs were relatively low in temperature as well, so that could be adding to kind of like the cooling effect to the overall study area. As for like water temperature, I mean, sorry, you said like air, right?
Yeah, is it like the water because the area one, or is it the source of the groundwater that's the bigger influence on whether they want to cite as a
I think it's actually kind of both. So there are three identified locations of where groundwater discharge is occurring throughout the farm. There are more locations of it, but we didn't sample at any of those locations. So I think it could be kind of a tandem between both the groundwater discharge having a cooling effect and also like warmer air temperatures warming the overall area. Yeah.
Other questions? Thank you, Brandon.
Yeah.
Yeah. Yeah. Good. You got it. Okay, our next speaker, Carla Esquivel. Unfortunately, her mentor cannot be here today, but that gives me the great pleasure of introducing her. Carla and I go way back, so this is a very monumentous day. Carla started in the UH system at Leeward Community College. As many of our students start at community colleges before they come in Manoa. And as in many of those cases also, I think she was initially hooked by taking 201, Ocean 201. It was being taught by Michael Lane. For those of you that have been around for a while, Michael was a student with Dr. Fred McKenzie in our department. So Carla transitioned from Leeward, came in to GES. She's had multiple research experience opportunities, National Marine Fisheries Service with NOAA, and also participated on a HOTS cruise. Carla's research experience with Dr. Yanagihara is regarding box jellyfish in the Philippines and awareness. And this is something that's very close to Carla because she is from the Philippines. And so I think this project had an extra special importance. So Carla.
Good morning, everyone. Thank you so much for coming to join us in our presentation. My name is Carla Esquivel, and the title of my research is Philippine Box Jelly Project, Educational Outreach to Raise Awareness, Reduce Injury, and Save Lives from Box Jelly Stings. And my mentor is Dr. Angel Yanagihara. So to begin with, why the Philippines and what is box jelly? So box jelly is found in the Philippines where livelihood, recreation, and culture of the people, especially of the coastal community, involves the ocean, much like Hawaii. And residents have been reported to have been stung by box jelly, and box jelly sting can cause extreme pain and even death. So what is box jelly? Box jelly belongs to phylum Cnidaria, and other animals that belong to this group are corals, Portuguese manipur, moon jelly. And the defining characteristic among all these, all the members of phylum Cnidaria is the presence of cnidae, which is a stinging organelle that is used for offense and defense. So how does the box jelly look? So the body of the box jelly has four corners, hence the name box jelly. And on each corner, tentacles arise. So for those belonging to class period data, there's only one, and then up to 15 in order Chirodropida. This one is a species here in Hawaii. It's called Aletina, and Dr. Yanagihara, my mentor, works closely with these. And an example of a multi-tentacled box jelly is this Chironex speckeri, which is common or also found in Australia and apparently in the Philippines. So all members of Cnidaria possess a penetrant cnidae called nematocyst, and nematocyst has a capsule, a hollow tubule, and venom, which is usually found inside the capsule. And when needed, the venom inside the capsule is injected through the hollow tubule underneath the skin. The hollow tubule can penetrate the skin like a hypodermic needle. And based on studies, among the members of Cnidaria, the box jelly venom is the most potent and the most deadly. So how can a box jelly kill? So the box jelly venom is a mixture of porins and enzymes such as proteases and lipases, small molecules, and venom lipids. And among these components, the porins or the pore-forming proteins is the fastest agent that causes death in the box jellies, in a box jelly sting. And this is according to peer-reviewed evidence-based research. So this is an example of the hole or the pores that is created by box jelly venom. Like, for example, when it was added to red blood cells, this is about 5 seconds after the venom was added, pores or holes starts to form. This is about 5 minutes after, in where everything that's inside the cell has leaked out. So this is an animation of that. This is an actual picture of the pore that was created by the box jelly venom, and the inner diameter is about 12 nanometers. And as you can see, compared to some of the components inside the red blood cells, like the potassium ion, which is about 0.28 nanometers, it can easily leak out of that, as well as the hemoglobin tetramer. It can also pass through. So basically what happens is when potassium ion inside the red blood cells leaked out into the blood plasma, it increases the level of potassium, which is a condition called hyperkalemia, and it generally stops the heart. So that what causes that is the cause of the death, the swift death of box jelly sting victims. And so why care? Injury and death caused by cnidarians is a public health concern worldwide and in the Philippines. And box jellies in particular are of overriding international medical significance because of their stinging potential to humans. So as I mentioned, there are box jellies in the Philippines, and these are like at least 18 locations that were identified to have 50 reported deaths. And this one, I wanted to say there's box jelly apparently, but it's not included in this. So it only means that there are even more, there's more places that box jelly exists. Sorry. So for example, these are actual incidents of box jelly stings. This is Ann Curtis. She is a famous Filipino actress. This was in the news in 2014. She got stung while filming her television series in this location. Apparently it's a box jelly hotspot. Fortunately, she survived. She received immediate medical attention. Unfortunately for these two children in 2018, this is an 18-month-old boy and a 9-year-old girl. Both of them died. She was in the news because her family was just visiting the country. So they died. And then also these are actual pictures of the box jellies that were shared like in the Facebook page. And these are, and you can see here, these are also found in Palawan, and Palawan is a famous tourist destination in the country. So with that, there is a collaboration effort between UH Manoa and some universities in the Philippines, which was started by Dr. Yanagihara. And the aim is to reduce mortality and morbidity caused by box jelly stings in the Philippines. And this network capacity building has 3 approaches: public health engagement, field ecology, and education and training. And this is where my study comes in. So the objective materials and methods is for me to create an education outreach material on box jelly in Filipino language based on peer-reviewed evidence-based papers, and to assess the awareness and attitudes towards box jelly engaged effectiveness of the said material on box jelly. So this is a screenshot of the first page or the first slide or the outreach material that I created. And this is the pre-presentation survey and the post-presentation survey that was created from this project. So the box jelly material, outreach material, it's in Filipino. It is about 12 minutes long. It was narrated by me. And there's about 23 slides. And the content is for information and awareness. So it's box jelly information, much like the ones that I presented earlier. And then sting management, which is equally important or more important. So under sting management, there are this portion emphasizes on awareness and prevention, such as prevention is better than cure. If you see a box jelly, please don't go in the water, put some signs out. And then the emphasis on protective clothing. And for first aid, there are 2 steps that are essential or that are crucial for this. And this is to prevent night-day discharge and to inactivate venom. So these are some of the slides that are included in the outreach material. And so the respondents for this pilot study were the students of Leyte Normal University. These are 7. So Leyte Normal University, Leyte is one of the locations of the box jelly stings, and Leyte Normal University is one of the collaborating universities in the Philippines for this project. And this was administered by Dr. Yanagihara, and these are her pictures that she shared with me. And so among the results of the study is that more people avoid the water if box jelly is present. So it seems that they are aware of the seriousness of this thing. So for example, in here, even pre-presentation or before the outreach material was presented, at least 70% said that they're not going into the water if box jelly is present. And another result that is that clothing at the beginning, they said clothing can actually help, or protective clothing can actually help prevent box jelly sting. This was 12% said, 12% believe that it helps. But after the presentation, at least about 68% said that that clothing can help. And as for the first aid questions, the use of vinegar to prevent night-day discharge and also to remove the tentacles, people will use it. Like the 52% prior to the presentation said that they will use that vinegar is what they're using to help with this. And then this increased to about 90% after the presentation. And also this one, the use of hot water, basically to inactivate the venom and to give relief also. Before the survey, only 24% said that they are going to use hot water, but after the survey, about 70% said that they are going to use hot water treatment instead of cold water treatment. So in conclusion and for future work, I believe that this outreach material has done what it says, which is to increase the knowledge and awareness on box jelly and the first aid also, and also the people are already aware of the seriousness of the box jelly sting. And then to make this outreach material accessible, like share on social media or places such as Facebook, Instagram, or YouTube, and also proof a proper survey and to administer to more groups for more data about box jelly and box jelly incidents in the Philippines. And so I would like to thank Dr. Yanagihara and Dr. Perkel, and also my Europe family, Ohana and GES family, and the Leyte Normal University students. And I would like to share this definition of outreach, which is an aspect of teaching beyond campus walls, an aspect of research that makes available the discovery beyond the academic community, and an aspect of service that benefits the public. Thank you.
Questions for Carla?
Okay, so. Next to the material is not the languages.
Oh. Okay, oh, did I mention, okay. The material is in Filipino. It's Tagalog, which is the national language of the Philippines. And I believe, if I'm not mistaken, that will be this Dr. A.L. Munower. He is from Indonesia. He was part of our group. And I believe that he and Dr. Yanagihara are planning to translate it to like Indonesian, to their language. Sorry.
For the native students.
Oh, I'm sorry, what?
Did you translate it to Wright for the native students?
Oh, no, it's in Filipino, in Tagalog only. And a lot of, it's a national language, so everybody knows, I assume, in the Philippines, because it's being taught as a course in school. Like you have to learn Filipino. Hi.
First, I want to say I love your presentation. I think this is like really important. And clearly it's working because you see your results. But I was curious, I know you selected this one university based on its location, having box jelly stings present. Did you select that one over other areas for a specific reason? And do you plan to expand this presentation to other affected areas?
I believe Dr. Yanagihara is already doing that because right now she's in the Philippines actually. And yesterday she told me that she was speaking to the students at the PUP, which is Polytechnic University of the Philippines. And I think she's going to another location today. So yeah, so the reason why Leyte Normal University was because it was in Dr. Yanagihara's itinerary at that time. So it was more like convenience because she was working with this universities already.
Okay. Last question. Are there certain communities that are more affected by box jelly stings than others? That were instrument and more affected in recreational swimming?
It seems like a lot, like it doesn't have any like socio-economic like groups or anything. So anyone who's in the water basically. And actually going into this project or before meeting Dr. Yanagihara, I didn't know that we have box jelly. We actually have another, apparently we have other jelly species in our place. And I thought that was it. I mean, that was like really, because I've been stung before, but box jelly is present in the country and I didn't know that people can die from it. And so, yeah, I guess anybody who is, everybody is like vulnerable to it because everybody goes to the water basically.
Yeah, great talk. I was curious as you went through kind of the different cases where in different locations where there had been stings, if you saw a trend in terms of the prevalence of stings, if there was an increasing trend or decreasing trend, just because of the relationship that certain jellyfish have to like the changes that changes.
That's actually one of the things that this study or this project wants to address. I didn't include it in my talk, but there is a measurement trap in the Philippines, like with box jelly stings in the Philippines, which means that there's no like proper data collection for it because it's not really considered. It's considered like an environmental accident. So there's no like specific statistics for that. And so because there's no data, meaning no action for the government or from that, so we don't know.
Question. I don't know too much about box jellies. It's a really interesting thought. Are the ones that are you went through like 3 different ones? Are the multi-tentacle ones more dangerous, like responsible for more deaths? And also, what is the response time that's required to prevent death from humans?
Like this one, response. Well, based on this, based on some studies, it says that like less than 5 minutes, I guess, because it says that I think the research was done. It says that by around 5 minutes, people can die because of the heart of the victim has stopped basically. So
Go back, yeah.
Thanks for teaching us all about this. In why do that? Predict when these box jellies are gonna appear, because we know it's related to like the food cycle, right?
Yeah.
I'm wondering if there are any ability to predict that for the species that are in the Philippines, the dolphins are going to be around as their problem.
I know that here in Hawaii, there's it's like every month, Dr. Yanagihara and her team goes to Waikiki to get some samples, and this is for the Alatina. In the Philippines, I don't think that there's any predictors for that. I mean, any effort such as that. But then again, one of the questions, sorry, of the survey, I asked the people if, I asked the respondents the months, like for example, the months that they observe box jelly, and apparently I'm not sure if it's true, but box jelly is being observed all year long, except September. So, and of course, it's very prevalent during the months of summer, like now until about July.
I have one question. Actually not a question, but just kind of get your gut feeling. So, with the surveys in Tagalog, and obviously there's some literacy element to that. How do you think, what's the best approach for outreach to maybe the populations that either can't be given the survey or may not be able to actually read the survey? How do you think can best?
Outreach, but okay, so another thing that Dr. Yanagihara is doing is she goes to local government units, like certain locations that she has established a relationship with, and then they help her. There are people who help her with that, and also I know that she is also presenting some materials prior to this outreach material. She's already doing that, basically. So.
Okay. Thank you, Carla.
Thank you.
So Madelyne is our next speaker, so I'll have Amber come up.
Hello, everyone. It is my pleasure to introduce Madelyne Harding. Maddie came to Manoa from Wisconsin on a track scholarship, where the 400 and 800 meter were her key events. In addition to being a student athlete and a GES major, she also has worked out of time as an undergrad, and she currently works at the Seed Conservation Lab at the Engineering Plant of Arena. And her long-term career goals are to continue her education and work in a way that contributes to natural resources management. And I got to know Maddie because she took my ecology course, which not a lot of GES students do, and I probably talked too much about lizards in there, but she got excited by that and came to work. And her project was looking at behavior in day geckos, the jewels of the Indian Ocean that so little is known about these lizards, and in social behavior and reptiles in general, that with Maddie's work, she has the expertise equivalent to anybody in the world in formally studying behavior of animals. And there's so many great things I could say about Maddie, but the best thing would probably be her genuine curiosity and enthusiasm for the animals and the system. She has told everybody about the lizards and the project, and it's been really great to see both her two. You need that curiosity in science to get you through the tedium and the many hours of video she went through these lizards that are mostly doing nothing. Pull out the little spots where they are doing interesting behavior. So it's been wonderful to see her hold that confidence to be able to become that expert and make those calls. What are these animals doing and why are they doing it?
Well, thank you, Amber. I am Maddie Harding, and I will be presenting my research on context-dependent behaviors in the gold dust day gecko.
All right, so just a little bit of background on social behavior. It's about fitness. Social behavior analysis helps relay information. It helps them build what genes they need to spread, territory, being able to relay information that they can't communicate like us. But unfortunately, not much is known. Now moving into the day gecko. It was illegally released in Oahu on the 1970s. It's formerly known as Lakavi. Oh my gosh, I got nervous. Sorry. It is originally from Madagascar and the small islands off of Africa. Then little is known about this day gecko. So our study questions were to describe the social repertoire. What are they doing? When are they doing it? Does sex depend? Does the frequency of behaviors differ based on sex? Are males displaying one behavior more than females? Does the frequency of behavior change based on context? Will one behavior be displayed more when male interactions are occurring or when male-female interactions are occurring? And do the different contexts occur in different seasons? So first part of the project, we had to build an ethogram. An ethogram is going to be describing all of our behaviors that are seen. I pulled from literature, so I based it off of anoles mostly, is what has been the most documented. And with that, I constructed, after hours of watching videos, all of the behaviors that were reoccurring and seen in the videos. Some behaviors were displayed and I found only in the day gecko. An example of how our histogram works is we'll have the behavior name, our description, and then an image. So for this one, the setup of this experiment was we went to Waimanalo, and with Wright's lab, they had set up 7 different enclosures that were 10 by 10. All of my videos were taken by Stevie Kennedy Gold during her thesis for anoles. With that, the enclosures all had the same plants in them, so they all had roughly the same setup as in here. And then observations were done on each gecko for 20 minutes, standing outside of the enclosures shown here. Identification of the geckos. We used, all of the geckos were collected in Manoa, and they were injected with the VID in each gecko here, kind of like a little tattoo, and they were sexed. And then each of the gecko had their back pattern printed here. Each back pattern on the geckos is very specific to them. It's like their fingerprints, so that we were able to use that to tell who is who. After watching countless hours of videos, I was able to go into the gecko pens and be able just to pick one up and be like, oh, I know who you are, and be able to say who and what that gecko was. So for our video analysis, we used Boris software. After every single video interaction was identified, if it was a male-male, if it was a male-female. We did have one video that was female-female, but only having one video, it was excluded from this research. Each individual was identified in the videos to know who was playing what part. And at first, we did an Excel spreadsheet to get all of the tallies to understand if there was a pattern in frequency. Was one gecko doing more behavior more than the other? And then we used Boris. So with our ethogram and Boris, we were able to assign each behavior, I should say, letter on our keyboard. And each gecko was either gecko 1, Kia 1, or gecko 2. So each one going through while the video is playing, do that. You would key what each gecko is doing. It took a lot of time and lots of going back to be able to key who is doing what in live time. Alrighty, and now on to our results. So we only had 4 male-male interaction results, but with them, we were able to find that they did only occur in our summertime. We had 141 videos in all of the day geckos, but only 64 of them contained interactions with the geckos, so there was more than one. And then with that, we only had 24 for female male were used due to having to exclude some for not meeting their criteria. Not both geckos were in the frame, the videos were not long enough, or the camera quality was just too hard to see and be able to understand what the geckos were doing. The interactions did have a peak time. We found that most occurred in the summer months here for both female male and male male interactions. And then in the male male interactions, we found that we have in every account that one of them will stop while the other one moves. There are 8 individuals in this, so we weren't seeing reoccurring pairs. And we also found out that the stalker does not depend on size. So in one of our accounts, the smaller gecko was in fact the pursuer of the bigger one, which was a bit shocking. And again, all of these occurred during the summer months. Now on to our male-female interactions. So with this, we used 22 behaviors out of our ethogram, all listed on the bottom here. The behaviors with the asterisks were deemed significant by p-test. Our first one are the ones that were pretty close in frequency. Both of them were exhibiting them. These are ones like eating, observing. They're going to do that even if there's not interaction. These were just background behaviors. Some examples of these are the curl, stretch, and the air leg, but is being a little blocked by the doing lid there. And then onto our B panel, we can see the behaviors that males dominated the most. And in these ones, the parallel stance, body glide, head bob, and stop were all deemed significant by p-test. This is super exciting to see because these are all very unique behaviors and fun to watch. Examples of these, the parallel stance is right here. They will line up facing what heads on other side. And then the stop. This guy is pursuing that one, and the body glide, where the male will cross over the female's top and relay some sort of information. And now this one shows our female dominated behaviors. In this one, they show the tail wag and the move being significant. One that I would like to point out, just because even though it was not significant, it was displayed the most in females, was our Yoshi tongue. The Yoshi tongue is a behavior that was found to be exhibited as of right now only in day geckos. Exhibit of this is the tail wag, so the tail will go in an axe motion, and then this is the move. This is an example of the Yoshi tongue. So some of my big take homes are the gold dust day geckos exhibit complex social repertoire. They have, I don't want to say personality, but they are so interesting. They are sassy. They will display their backs in just full sorry, I could go on forever about that and I have to watch my time. But they are very, very complex. All of these behaviors are likely part of a courtship function. This could be relaying pheromones. It could be relaying size. Are we related? Are we a match? Have we mated before? All of this is being relayed in a few simple actions that are happening, as you saw in the video, very, very quickly, just as I'm communicating to you. And we found that the occurrence does have a peak, which is amazing because it coincides with research from another student in Amber's lab that found that juveniles peak in the fall and spring months, which helps support the evidence that this is sporty behavior. Males and females also behave differently. They have different repertoire. They do different things in different situations compared to each other. And all of this can be looked at deeper in further studies. If we put two known individuals together, are they going to display more? If we put two unknowns together during different times of the year, will we get to see more of different behaviors? With that, I would just like to take a moment to thank my mentor for everything that she has done. And I would love to thank Robert for taking the time to read my thesis. And then of course, Tina for answering all of my emails. They were a lot. I would also like to thank Jeffrey Drazen for taking countless hours with me to form my schedule, listen to my grievances about my schedule, and assure me that everything was going to be okay with it. And with that, are there any questions?
One question is, these are all baseline footage.
Yeah. Yeah. So the day gecko, they're diurnal, which means that they behave mostly during the day. Because there isn't a lot known about them. We're not really sure what they behave at night. I believe that there is a observational paper that was done on Big Island discussing what they react at night by lights with, and they were just on Big Island kind of looking at them, so it was basically just observations. But other than that, not much is known about it. Sure.
Because their behavior seems to be portrait related, did you know like a time difference between male and male interactions and female interactions? Are they following male-female interactions?
So with the videos, they were all taken for Phoebe Kennedy Gold's thesis project. So it was more of like as soon as they observed a gecko, they would start filming just that one and then follow that gecko. That also added some difficulty when I was going through all of them because it was just focusing on that gecko, so you couldn't always see what the other one was doing. And that's part of why some of the videos had to be so good.
So there was an increase in kind of overall behavior during the summer. Did you see any kind of behaviors that were only exhibited during other seasons, or like an increase in certain behaviors?
We haven't, but that would be really interesting to look at. I know that we've only seen male-male interactions during the summer months, and then female-male interactions dropped during the summer, fall and spring months. So, I mean, it's definitely possible that they could be displaying other the behaviors during that time. We just weren't able to like go through and view it. But that would definitely be an interesting next study.
Yeah.
Is it known whether these things are territorial? If they are, if some of these behaviors appropriated to them?
Yeah, they definitely are. I would say that they're not as territorial as anoles, because the anoles seem to always be fighting compared to these guys. I think attack was what only happened a couple times in all of our videos, but most of it's like the stop and shake type of behavior to get them out of potentially an area.
You interested in looking at the other data species, giant data, smaller advanced.
Yeah, that would be super interesting. It's just difficult here as the giant day geckos are less common and are illegal to bring in. So I would be a little bit hesitant to do that study here at least, just because of that. But yeah, that would be really, really interesting to see how it correlates over the two species.
Go ahead.
Thanks for sharing all of that. You mentioned that like you started to get an idea of the individual geckos, and I was wondering if you noticed specific interactions happening between a certain pair, like you notice that they have friends or
Yeah, two that really come to mind are Plat 102 and Plat 104. We have a lot of videos with them interacting. I'm not sure if that's just like they were the most active, that was like their personalities, they were running around, but those were a significant amount of videos that we've seen in the data. Yeah.
Alright. Okay.
I live in the back of Lowell Valley, and I would say my house is dead gecko central. There's a huge gradient on the outside on my traveler's palm, inside where I have broken populations that we encourage the marmorated stuff.
Karen, does any of this have to do with you guys' hot tub or?
I also love putting that I feel guilty about encouraging them to are an invasive species, or can I just relax into my pets? I don't really have to take care of them.
So because so little is known about them, I'm not sure how they're affecting like native species, as they are an invasive species that there's going to be costs and benefits. I'm not sure if those costs and benefits are truly known right now. I mean, they could be harming the endemic insect population, but we're not sure if they're crossing over, as there's not research on that. I mean, they like ants. They constantly were looking at that. So, I mean, that's always a good thing to get rid of those in your house. So I would say right now, until further research, that it would be okay.
Okay, last question.
So fascinating, your research. Thank you. So I have a couple questions. So geckos were in each enclosure, and then was all the video footage from the minutes of people with the camera, or was it ever just that alone? And then the other question was, did you have anything? Did you find much literature about behavior that they?
Yeah. So each enclosure actually they had different treatments. So some had the two different anoles in them, and then some were just pets. So all of them were done by like the natural gradient of like population diversity. One of the things that we talked about was potentially looking at if some of the enclosures with the anoles had different reactions, but because they have different substrate levels, we weren't sure if that would really. Then your second question was. Oh, yeah. So all of them were from observation standing, but it would just that way we could make sure to follow the gecko because they are small and they blend in really well with the substrate being so green. It would, I think, be hard to like set the camera out and just watch them and have the chance of them coming back and forth because the observations were so limited, just being able to spot them. And then your third was about the literature. There was a few papers, but it was more of like in passing, it was discussing their anatomy. There wasn't any really about the behavior at all. It was just kind of discussing their anatomy, or this is what they like to eat. It wasn't ever describing their behaviors.
Thank you, Hannah. So our next speaker is Kyra, and Bill, if you could come up and.
So I have the honor of introducing Kyra. She came to UH from Portland, Oregon. She grew up on the coast, sailing and doing ocean athletic studies, marine studies here. So since Kyra has been at UH, she's really done nothing but excel. She's on Dean's list consistently. She will graduate with a honors certificate. She also got a Noah Hollings scholarship for her time here at UH. That included an internship where she got to work with scientists at NOAA, where she worked on getting estimates of sea ice thickness from satellite data. So she's really done a lot already. And she also participated at the GES here and helped oceanography faculty plan a symposium for graduate students or PhD graduates from around the country. And then, working on our thesis, which you'll see here in a moment, it's really been a pleasure. So she's very diligent, always on time. We brought a lot of her own ideas to the process, which is really refreshing and really enjoyed the programmer actually during her time. Kind of started with some basic knowledge of Python over being a long way. So yeah, here.
Do you guys know how to go into presenter mode? All right. Hi, I'm Kyra, and my mentor is Dr. Phil Thomson, who introduced me. And my project is quantifying the transition from occasional to chronic coastal flooding. So sea level is rising globally. This is due to climate change, and it's because as the Earth's temperatures are warming, there's increased thermal expansion in the ocean, and then also melting ice on land that's flowing into the ocean and increasing the mass of water. This plot is a projection from the US government used for planning purposes of the future global mean sea level until 2050. And as you can see, they use a few different scenarios. I think it's important to note that the highest scenario is extremely unlikely because that would require a significant increase in our emissions. But the low scenario is also unlikely because that would require a significant reduction. So the scenario I use for most of my analysis is the intermediate one, and that reflects more of a business as usual emissions scenario. And then sea level also varies regionally from this global mean, and this is due to geologic and oceanographic processes. So as sea level is rising, we can expect to see a lot more coastal flooding. The increasing sea level is the primary reason for these increases in coastal flooding, and they're driven both by storm surge and by high tide from the natural tidal cycle. Although as sea levels are rising, high tide only related coastal flooding events are also increasing. So this is a plot from the US, and it shows just observances of flooding throughout the years. And you can see that they have been increasing, and this trend is projected to continue. So there's already a lot of really vulnerable low-lying coastal countries that have large coastlines and are already susceptible to sea level rise issues like storms, erosion, and freshwater scarcity, all things that will be exacerbated by increased frequency of coastal flooding. Planning for those large flooding events that we see in the news, the destructive extreme ones, is important, but there's also a lot of damage from smaller events that occur more frequently with less time in between to repair any infrastructure or other damages that might occur. So most global studies thus far have really focused on the amount of sea level rise required for a change to reach high tide flooding, but they don't offer a time scale for this transition. And that's the most important part because that is what is needed for planning, and that is what's tangible to people like, us, the general public. It's a lot easier for us to imagine the number of years until we can expect to see flooding than it is for us to imagine, an increase in centimeters or meters. Which brings me to my research questions. So how much time is there between the onset of coastal flooding and chronic coastal flooding? And then how does this transition differ across the globe? So I used a global array of tide gauge data, and each tide gauge, in order to be valid, had to have a tidal record of more than 20 years with more than 320 days for each year. And then we detrended the tidal record for each of these locations which was valid because we're more interested in the change between sea level as it rises and the change between extreme water levels than we are the overall upward trend. It also is okay because the variation within each individual year could theoretically reflect the variation of any past year. So we define chronic flooding as flooding that occurs on more than 20 days per year, and occasional flooding as flooding that occurs only about once per year, so less frequently. And from my detrended time series, I calculated the change required to reach chronic flooding in centimeters, and I called that my delta H value. So laser there. Oh, okay, never mind. So the blue line is the previous tidal record, or it's showing the daily maximum sea level. And then the orange line is that same tidal record, but with the delta H added. So it's raised by delta H. And the thick black line on the top is our flooding frequency threshold. And as you can see, the blue line only exceeds that threshold a few times, whereas once that sea level or the daily maximums are raised, because the mean sea level is raised, then there are a lot more frequent occurrences of flooding. So that orange line exceeds the black one much more frequently. So then using my delta H value and some projections of future sea level rise until 2100, I got my transition time, which is delta T. And transition time is a measure of how soon we can expect to see chronic flooding. So shorter transition times means we can expect to see flooding sooner. Okay, sorry, let me move this. So we did this analysis both starting in 2020 and in 2050 because we can expect to see pretty extreme changes, especially increases in the global mean projected sea level starting after 2050. And we wanted to capture this change in our analysis. However, the year right now is 2023, so our projections starting in 2020 are a lot more relevant, especially for intermediate planning purposes. So that's what I'm going to focus on for the rest of this presentation. As you can see that there is a pretty wide range in values for delta T, ranging from 16 to 130 years. And then there's also some spatial structure to my results. So the higher latitudes have those dark purple values. These are a lot longer transition times, whereas lower latitudes tend to have lower transition times. So for example, here in Honolulu, we can expect a transition time of about 21 years, which means we can expect to see chronic flooding in 21 years or less. And a lot of the spatial structure is likely due to increased storminess in the higher latitudes compared to the lower latitudes. They're just stormier in general, and this leads to higher differences between the extreme water levels from storminess versus just the mean tide. So as that sea level is rising, there's still a difference there. Okay. So here are my results grouped by region. The gray line in the middle of each plot is the global median, which is 31 years. And I think the most important result here is that islands, which are the red dots on the left, are significantly lower than the global median. So islands have a median of 25 years. And I think this really indicates that these are areas we need to prioritize when we are thinking about addressing flooding in the future. I think especially compared to Northern Europe, which is the region with the largest transition time of 62 years, islands have less than half of the amount of time that Northern Europe does. And I think that just illustrates how much more we need to prioritize these islands over, other areas. So we were also interested in what dictates the length of the transition time. So we removed the part of the tidal record from the that was not due to the natural tidal cycle, and this is what we called our non-tidal residuals, which are equivalent to storm surge. So on the bottom plot, you can see the standard deviation of the non-tidal residuals plotted with the transition time, and the top plot is the standardization of the natural tidal cycle with the transition time. And the relationship between the non-tidal residuals and the transition time was a lot stronger than with the natural tidal cycle. And this illustrates that the relationship, storminess is really what is driving these transition times. And even though, again, large stormy events have a lot of attention, both in research and in the media, the places with less storminess are actually where we're going to see much faster transition times. So you can see that, you know, all of these green and yellow values down here. And many of these places are also islands. So some of the limitations of the study is that we did not really consider the nodal cycle, which is the changes in the tidal cycle like on 10-year periods. And this could influence the transition time for certain locations. And we also didn't take into account any changes in storm frequency or severity in the future due to climate change that might alter the relationship between non-tidal residuals and transition time. All right, so global mean sea level is rising, and flooding is expected to increase in most places with this rise. If you have no other takeaway from this project, know that islands are the most vulnerable. They have small tidal ranges and lower contributions from the non-tidal residuals. And then most importantly, this transition time can be used to help predict and prepare for chronic flooding, but locations with the shortest times need to start addressing these changes now before they occur. So I would like to thank my mentor, Dr. Phil Thomson, and then also Dr. Brian Powell and Dr. Glenn Carter for helping with this presentation and my thesis and reviewing it. Thank you.
Questions?
That was great. The figure you have with like the time and like the height change of the different like minutes or where you said the islands were close to the but it's super cool. So I was interested about like the Western North America and the Northern European, like the lowest heights. Those bars have like they have a lot of variability too. And so I was wondering, like, those points are already experiencing like other tidal, rise and coming with that. So is there any other parameters that you think are important for dictating what those locations are really vulnerable or why there's so much variation?
So the biggest thing is the storminess, and also because some of these locations have really wide tidal ranges. So there's a big difference between like, the occasional like, high water level and just the median and also the lowest. The other thing is that like, if you look at this plot, there's these yellow values up here. And that's actually where sea level is decreasing because as the glaciers and ice that's up there on these land masses melts, then the land actually has like an elastic response. So it's rising. And then relative to that, the sea level seems like it's getting smaller. So that is relevant to these yellow locations. Then it's also relevant to kind of like up here in Alaska and then some of these areas in Northern Europe, because glacial isostatic adjustment is happening there too. But then we can see a faster rebound that will actually lead to like quicker transitions after all of the land is done, rising.
Jim.
These long-term trends are coming from climate change.
Yeah, they come from the NOAA Sweet et al. Report on things. You mean these ones?
Right.
Yeah.
So I'm just wondering if you're confident in the parameterization of islands in these climate models and whether that's going to throw some uncertainty in that.
So these are, it's just a gridded data set. And then I just took out the same location for where the tide gauges were that I used from that gridded data set for islands. So I'm not sure about like individually how they calculated those, but I'm pretty confident in it, I guess.
Yeah.
Hi, Kyra.
Hi.
I just like to know, from your experience, how did that relate to your project and how, what you learned at NOAA, how to do this project?
Well, I guess at NOAA, I worked at the satellite operations facility, and I didn't really use any satellites in this. So nothing, I guess. I got better at Python, yeah.
You're looking at continental as an application. That more significant than the difference. Like you say, based on latitude, like is island versus continent more important than like, location?
Well, so they are also related because many islands are at lower latitudes, especially the ones we have tide gauges for. I think that they are more relevant because how islands are, like these are low-lying, I guess, like I said before, and many of them have you know, less access to resources and have to import a lot of things, and are just overall a little bit less prepared for like these large climate changes. Whereas continents, you know, they have like trees and concrete and stuff inland that they can get. So I think it's more relevant just contextually, yeah.
Great.
It's a lot of problems that we mentioned are also a lot of those countries here now, either islands are island countries. And because of the relationship between the residual analysis with the tide versus storm surge, like how those were made. And I noticed that like the mid-latitude areas, or low areas, that was less of a difference. Which model fit the data better was one was much more superior continental areas, maybe not for islands. So I'm going to ask you about these East Asian countries, Indonesia and the Philippines, that are in areas where sea level rise is, you know, I don't really know if this is a big of a problem. And they have, you know, they're close to continents rather than being out of like island areas. So I'm wondering if you like conjecture about how these countries are going to be impacted, given that they're these countries that are kind of in maybe biased area.
Well, I guess first of all, I will say that we kind of defined islands as like a little arbitrarily because it's like there were questions like, is New Zealand an island? You know, like technically yes, but also no. So things like that are also islands that are close to land, like the Aleutian Islands in Alaska. So those things are a little bit objective. As for like how the Philippines will respond, like in their response, like planning wise, I'm not really sure because the planning part was outside of the scope of this study. But I can just tell you that the transition time for, you know, these areas tends to be about like 15, 20 years. Yeah. Sorry, that wasn't really
No, that's good.
okay.
Thank you, Kyra.
All right.
Okay, our next speaker, Tehani, will be introduced by Dr. Dulai.
It's our great pleasure to introduce Malterre, who was born and raised on Oahu and has a lifelong passion to care about the environment. Therefore, it was logical for her to join the GES program in 2019. And specifically, she greatly values the opportunities to study environmental issues here in Hawaii, because when she graduates, she hopes to continue in that, and they continue her passion and also the profession. Also, as you will see based from her presentation, she went into working with a community, start with a hypothesis. The hypothesis did not get confirmed. Good for the community that there is no environmental issue yet. How do you present about something that you were really hoping to do some cool analysis on? Well, Tehani did a great job with that. And so you'll get a pleasure of seeing that. I would also like to mention that after the graduation, she will continue on as part of the 4+1 bank to the continuation program for GES. Get a master's in finance. Please welcome Quinn.
Okay. Hello, everyone. My name is Tehani. Dr. Dulai, who just introduced me, as my mentor. She's from the Department of Earth Sciences. And my project is looking at potential threats to water quality at an urban wetland farm here on Oahu. So a brief overview of my presentation today. I'll be starting with an overview of my study site and current concerns that the farm managers are facing. I will then go into pharmaceutical water tracers, which are the substances that I analyze in my research. I will then go into the methods that we use, and finally our conclusions. Okay. So a brief overview of Sumida Farm, which is my study site. As many of you may know, Sumida Farm is a watercress farm located here on Oahu. It is specifically located in the Awaya area in the Apua'a of Kalawao within the Moku of Eba. The farm was first established in 1928, and since then it has been operated by the Sumida family, making it one of the last remaining multi-generational family-owned farms here in Hawaii. It is also a really important part of our local agriculture since it produces around 70% of the watercress here in Hawaii. And lastly, the farm provides immense community and ecosystem services. So since the farm has been in operation for nearly 100 years and it has persisted in this area that has otherwise become highly urbanized, it has really immense value to the surrounding community, and school groups go there for field trips to learn about local agriculture. Additionally, it kind of acts as sort of a wetland within this urban setting, and specifically, it can provide habitat for some of the birds that you can see in the picture, including some native species such as the Io. So a brief overview of the layout of Sumida Farm. There are 111 plots over around 11 acres on the farm. As I mentioned, it is in the middle of a highly urbanized surrounding area. So in this image, you can see that Pearl Ridge Shopping Center is actually immediately north of the farm, and Kamehameha Highway and the rail system is immediately south of the farm. And Palimomi Medical Center is also nearby. So for the farm layout, the farm receives the majority of its water from freshwater springs on the Mauka section of the farm. Two of the major springs are highlighted in the blue dots, but there are also multiple smaller springs that help provide water to the farm. So after the water flows from these springs and through each of the watercress plots, it is then returned to the surrounding watershed through these two outflow points. So understanding the water quality at Sumida Farm is also important for understanding what type of water is going back out through those outflow points. So some of the concerns that farm managers are currently worried about. The first is proximity to sewer lines and the surrounding urbanization. So in this figure, you can see the sewer mains that are adjacent to the farm indicated in red. And as you can see, there are a number of them that are adjacent to the farm. These can become sources of wastewater contamination if there's any leakage in this infrastructure. And the next is how climate change will affect conditions at the farm. So thank you, Kyra, for introducing everyone to climate change in your presentation. But primarily, it is known, some research has shown that things like increasing storm frequency and sea level rise has the ability to make our wastewater infrastructure here on Oahu increasingly vulnerable. Specifically in the area adjacent to Sumida Farm in this red circle, you can see some of those colored dots, which are wastewater infrastructure that are expected to become increasingly vulnerable under a 3.2-foot increase in sea level rise. So the goal of my project is to determine whether there's any wastewater present at the farm, specifically by testing for two pharmaceutical wastewater tracers, caffeine and carbamazepine. So what are caffeine and carbamazepine? As we know, caffeine is a substance commonly used, and it is found in coffee, tea, chocolate, energy drinks, and different medications, and it is really commonly consumed and excreted by humans. Carbamazepine, on the other hand, is not as commonly consumed and excreted, but it is a chemical compound that is found in anticonvulsant and mood stabilizing medications used to treat conditions like epilepsy and bipolar disorder. Overall, both of these substances are excreted by humans and can persist in the environment, making them useful tracers of wastewater. So if you find these substances in the environment and in the aquatic environment in particular, it can be indicators of wastewater contamination. So for our methods, our first step was collecting monthly water samples. So we had 10 sampling plots from around the farm, and samples were collected monthly over the course of 11 months. Our second step was then analyzing our initial results with respect to various detection limits, which I will go into further in my presentation. And the last step was finding if there's any correlation between caffeine and carbamazepine presence at the farm and rainfall. So the reason why we wanted to do this is because, as I mentioned, one of the projected impacts of climate change is increased storm frequency and intensity. In addition, rainfall can also increase runoff that can facilitate the flow of these contaminants and wastewater onto the farm. So our first step was monthly water sample collection. Over the course of 11 months, we collected 122 total samples from the farm along in these 10 sampling plots. We then analyzed our samples in the lab with enzyme-linked immunosorbent assay or ELISA kits. And in our initial results, we found that 25 out of the 122 total samples contained some level of caffeine, while 3 samples contained carbamazepine. However, these initial results were based on the manufacturer's reported minimum detection limit that was created in ideal lab conditions, which is not the conditions that we analyzed our samples in. So we then analyzed these initial results with respect to various detection limits. So as I mentioned, we had those initial results that were analyzed in accordance with the manufacturer's reported detection limits developed in ideal lab settings, which is different from the settings in which we analyzed our samples. So in order to see if there was any significant difference in our analysis, the first step was creating standard curves. So standards are a set of solutions that are provided with each ELISA kit in which there's a known concentration of caffeine and carbamazepine. So once you have those standards, you can then plot them against your samples to see where the samples line up within those concentrations. So when you plot your standard curves so for concentration versus B over B naught, which is the mean standard absorbance to the zero standard absorbance ratio, you want to see something like this for your standards. So the blue dots are the standard solutions, and they kind of line up nicely along this line or the standard curve. And this is what we see in our carbamazepine kits in the second round of sample analysis. We got a really nice standard curve for that round of analysis. However, for the caffeine kits, they really didn't perform very well. The blue dots, as you can see, are scattered, especially in this section here. And what this means is that within that range of scattered samples, we can't really be 100% sure that the samples within this range really did contain the caffeine amount that was initially reported. And within that range is 1 part per billion and below. So after creating these standard curves and comparing our samples to those curves, we applied various detection limits, namely 90% and 50% B over B naught. So as I mentioned, B over B naught is the mean sample absorbance to the absorbance in the zero standard, or the standard with a zero concentration of caffeine and carbamazepine. So when you're applying these different detection limits, it's a way to say, as you narrow down these results, you can say with increased confidence that these samples really did contain caffeine or carbamazepine. So when we look at our initial results compared to when we applied these various detection limits, you can see that it really got narrowed down. In this study, the manufacturer's detection limit was the least conservative approach that was used,
while I-50 over here, or 50% B over B naught, was the most conservative approach that we used. So what this means is that we can say with the most certainty that these two samples really did contain caffeine, while there were no samples that contained carbamazepine under these detection limits. And this is a really great sign for the farm. So using this data, we then tried to compare it to rainfall. So precipitation data were collected from the nearest weather station, which is around 4 miles away from the farm. And this was our results for that precipitation data. So the daily precipitation throughout the study period is indicated by the blue dots and lines, while the precipitation on our sample collection dates are indicated by the orange lines. And as you can see on the dates that we collected our samples, there was very little, if any, precipitation. And this proved to make it hard to determine any correlation between these substances and precipitation. Overall, since after we narrowed down those caffeine and carbamazepine results, there really wasn't very much at all. In addition to the lack of rainfall on or before our sampling dates, there wasn't too much to compare against one another. And we didn't want to sort of force a correlation when there wasn't many data points to compare against. So this could definitely be a point of future research. So our conclusions. The main conclusion is that there is little to no caffeine and carbamazepine present at Sumida Farm throughout the study period, which is a great news for the water quality at the farm and lack of wastewater contamination. However, this could be influenced by the fact that carbamazepine is not a widely used substance to begin with. For example, if it is not commonly consumed by people in the area, it then won't be present in the wastewater to begin with. So to kind of account for that, this is the reason why we had 2 substances rather than just one. And as I mentioned, caffeine is much more widely used than carbamazepine. As I mentioned, the caffeine kits did not perform as effectively as the carbamazepine kits, and thus we could not determine if the samples at or below 1 part per billion of caffeine were really what they were reflected to be in our results. Overall, more research is needed to confirm any correlation with rainfall. This could involve maybe studying the samples over a longer study period, or perhaps taking samples more frequently so that they could fall in line with different precipitation events. And overall, the study can serve as a baseline for current conditions at the farm. So as I mentioned, conditions are expected to change in the future under climate change, and our wastewater infrastructure can become increasingly vulnerable, which can increase the chance that wastewater can then be transported onto the farm from runoff. So this is, if for future research, if the study is continued at Sumida Farm, this can serve as a baseline for current conditions in 2022. So this work was supported by the National Science Foundation and the Hawaii Pacific Foundation. I would like to say a huge mahalo to my mentor, Dr. Dulai, Dr. Arigato, who helped to review my thesis, the Sumida Farm managers, and all the student and faculty researchers who supported this project and made my research possible. Thank you very much.
No worries, I don't know, but if there's water present, is there possible bioremediation where
Yeah, that's a great question. So some of the research that I have found is that that's not something that's very widely researched. There was some research in these wastewater tracers being taken up by plants like cucumbers and other plants. And in those studies, they were able to take up some of these tracers, but the setting of those studies was really different than the setting at Sumida Farm. For example, the plants in those studies were exposed to consistent high amounts of these tracers, which would be much higher than what we would see at the farm. And they're also grown in a nutrient media, while this watercress at the farm is grown in just water. So I wouldn't say that's something that's very significant concern that would influence the data.
You're doing a great job. Okay, so you mentioned that carbamazepine and caffeine persist in others. They also persist in salt water. You were talking about climate change. You didn't mention the possibility of salinity, but yeah, do they also persist in salt water?
That I'm not actually too sure. I know carbamazepine is much more stable in the environment, so it has a much longer half-life than caffeine. So I would assume that it is likely to persist still. These substances are degraded under like photodegradation and biodegradation, things like that. So I would say, if I had to assume, they are still able to persist, but I would definitely have to look into that more.
I'm curious, when the campaign, what did you make your standard curve and like what solvent did you use? Was it just water or like?
No, it was I forget what the name of it was, but it was the things that came with the kit themselves. I think it was, I don't remember the name of it exactly.
And then for the standard curve, did you like do a dilation of like where you take the highest concentration standard and then like half, or did you add the standard to like a smaller concentration of the standard to different volumes?
So we kind of had just analyzed the standard solutions at the same time as our samples. So we didn't really go into depth with anything like that. They were just analyzed at the same time. Yeah.
Like I was like really curious about how your standard curve turned out to be like
yeah
for the place like that, and just trying to figure out how that happened.
Yeah, I mean, that's something that we're kind of wondering too. It could be due to a number of factors. It could be just how they came from the manufacturer or improper storage. So we didn't really go into depth with analyzing where those potential sources could have been. Yeah, we just know that, well, another thing is too, there are other methods for analyzing caffeine in water samples, but they're much more costly. So that's why we didn't use those in this study.
Yeah, it's really too bad about caffeine. It seems like an interesting like to work on in the future. Is there like your analysis are all in the PPP range, but is there a sense if there was like a big wastewater problem here or a different site, if they were using signals at that level, or would they be a whole lot higher?
At a different site on the farm or somewhere else?
Yeah, if you were just going to go around and say, like, oh, is there wastewater coming in here? It's what level of caffeine you say, oh, there's a big problem versus something or maybe a tiny amount, not huge.
That's a great question. I think, well, so caffeine degrades really quickly. It has a half-life of about 1.5 days only in the environment. So I think it depends highly on the environment. It could also depend on when that discharge happens. So if it didn't happen in a short time frame from your sample collection, it just wouldn't be there. So I think it could vary a lot really based on also your proximity to the wastewater. But I think the thing that we focused on the most in our study was quantifying which plots had the most and also whether it was a positive detection or not. So not necessarily focusing on those exact amounts, but seeing whether or not it was present to begin with.
Yeah, great talk. Wondering about the site you decided to work on, and if there was a reason to expect sort of or the fight with the site, or it was sort of.
Yeah, so those sample sites were chosen based off of concerns from farm managers. So past research has shown based on some different water quality parameters, there were concerns in certain areas of the farm. So we targeted our sample sites based on those concerns specifically, and also for land use in areas surrounding certain areas of the farm.
More questions?
Wondering, well, like I don't know much about how you analyze wastewater and context, but why those two substances? Like what is so specific about those two? Like is it the ease of use, the accessibility of those kids, or?
Yeah, that's a great question. So these substances are two of the pharmaceutical wastewater tracers that are commonly used for research of water quality here on Oahu. So in other regions, people have sampled them and found them to be concerned, but they're really common here on Oahu, and caffeine especially, it's pretty common everywhere. But yeah, that's kind of why we selected these two. And also why we selected them in contrast to one another is again, because carbamazepine is not very widely used, while caffeine is, and they also have different persistence in the environment. So I feel like sampling these two specifically kind of gave us that nice comparison to see how those different factors could influence them.
Thank you, Tehani.
Thank you.
Okay, so Quinn will be our last speaker, and his mentor, Dr. Robert Thomson, will be introducing us.
Hi, everyone. I'm happy to introduce Quinn Moon, last speaker in the session. Quinn came to UH from New Mexico, where he grew up. Growing up there, he spent a lot of time outside developing interests in conservation, management, and restoration of biodiversity. Since coming to UH, he's really taken those interests and applied them in sort of a stunning array of things he's gotten involved in, both in my lab as well as elsewhere on campus. So that involves things like experimental restoration work for forests on the Big Island, to herpetological work in my lab. So with us, he's been working with us for a couple of years now. He's gotten involved in this project focused on whip-tailed lizards, which have sort of captured the attention of evolutionary biologists for the unusual way that speciation happens in them. So Quinn's work has sort of involved a lot of different skills he's had to piece together quickly, from interfacing with a bunch of museum collections to manage a large set of loans, to doing some moderately tricky high throughput sequencing, to bioinformatics skills, to the statistical analysis skills, pull it all together. So today we'll be sharing his work on that with you.
All right, great. Thank you, Bob. Thank you for the introduction, and thank you everybody for being here. Today it's my pleasure to present on my undergraduate thesis, which looked at a population of lizards in Michigan to determine if they are native or introduced. Some introduction and background on the topic. Across the globe, reptiles are increasingly threatened with extinction. Some of the principal drivers of reptile decline include habitat fragmentation and degradation, as well as the anthropogenic or human spread of reptiles. These same drivers have also created a number of isolated populations with unknown origins. So these small and isolated populations are either the remnants or fragments of once continuous populations that are worthy of special management and conservation, or they're the result of a recent human-mediated introduction. In this field, genetics, or the field of conservation genetics, are increasingly used to sort of address these questions and answer them. This type of research is incredibly important for managing and delegating conservation resources, further investigating the impacts of invasive species, and importantly, locating and protecting these vulnerable native populations. For my study, I looked at 6-line race runner lizards in particular that are found in just one park in just one county in Michigan. The population is 322 kilometers separated from the rest of its range. So 6-line race runners have a pretty broad range spanning from Florida in the east to Montana in the west, but this population in Michigan is pretty separated from the rest of its range. The population was just identified in the late 1980s, and as I mentioned, it's just found in this Murphy Lake State Game Area. There have been past studies sort of trying to investigate if they're introduced or not, and these have all been inconclusive. This type of research is important as 6-line race runners are one of just 2 lizards that are found in Michigan, so it represents half the lizard biodiversity in the state. Despite the sort of confusing origin of the population, they're currently listed as native and protected in the state. We built off a report that they might have been introduced directly from Missouri into Michigan. So our results sort of investigate if this population is introduced or native. For the study, we built on the idea that the height of the last glacial maxima, the last ice age, glaciers expanded southern enough to cover the Great Lakes region and covering areas of Michigan, as you can see in this figure. So the population probably did not persist through glaciation in Michigan, but it could be the result of a post-glacial rapid expansion. So when those glaciers retreated further northwards, did population of race runners naturally re-invade Michigan, or were they introduced by humans into Michigan? So for the study, we proposed to use phylogenetics and sort of set up 2 possible scenarios. One is, if the lizards are native to Michigan, then we would expect them to group most closely with the closest known population, which would be Indiana. This would represent that the Indiana and Michigan population used to be a once continuous population that has since been broken down and fragmented, and they only persist in this park as sort of a remnant of a once continuous population. or the population in Michigan is the result of a recent human-mediated introduction. So we expected that on a phylogenetic analysis, we would see Michigan samples grouped most closely with populations that aren't from Indiana, perhaps Missouri, testing the sort of hypothesis that was presented by somebody else. For this study, we tried to collect as many samples as we could from throughout the range. So I relied on museum samples or samples that exist in natural history museums that can be loaned out. The green is a known range of the population, and the red dots are the places that I was able to sample and collect samples from. So the idea was to collect samples from throughout the range, sort of to test this, are they more closely related to Indiana or from somewhere else in the range? Some important sampling to note is that we were able to collect 4 samples from this sort of Indiana region, but there were no samples that were available in natural history collections for Missouri. So we'll talk about how that impacts our results. The first part of the methods was collecting all these samples from museums. So they arrived in the mail, either frozen or stored in alcohol. And I performed a pretty standard DNA extraction from the spin column and quantified the DNA. The next step, sort of, our effort were to collect regions of the genome from throughout the entire genome, rather than just focusing on any singular gene. So we relied on a DD-RAT approach, which sort of makes a reduced representation library of the whole genome down to just a select set of genes that we are interested in, which for our study was approximately 20,000 loci or genes. The next step was we sent the samples off for sequencing at the University of Oklahoma on the type of sequencing known as Illumina. Following the return of the sequences, we align them to a reference genome. So there's no reference genome for my species of interest, but there's a reference genome available for a species in the same genus. So I use that reference genome to sort of compile the DNA sequences, which is sort of a way of lining them up in a way that's compatible for a model. Following alignment, the next step was to do the phylogenetic inference that we were after. So phylogenetic inference is essentially a model fit to say, based on the DNA sequence data, what is the most likely evolutionary history of these populations? And then after sort of running these models to make phylogenetic trees, we can do bootstrapping, which is a way of subsampling the data to see how frequently the subsamples of data sort of agree with our original interpretation. And so these values are generally, at least for my example, I bootstrapped 100 times. So they're between 1 and 100, which is sort of a test for how robust is the model. So jumping into the phylogenetic results, the first thing to mention is that, which is the first result, that not every DNA extraction worked perfectly, and that I lost, or 8 of the samples that I originally collected did not have enough DNA to be included in the downstream analysis. So that was particularly important for my Indiana group, in which of my original 4 samples, only 1 had enough DNA to be included in the library. So the first thing we notice is that on our phylogenetic tree, that the topology generally reflects geographic location, which makes sense. Things should be most closely related to things that are from the same place as it. And the first result that comes to mind is that the populations are sort of follow this general trend of southern populations being the initial or earliest populations, followed by a sort of a general northward expansion of the population. So we can see that our most derived populations in Michigan and Indiana or the most northern populations are sort of descendants of southern populations, which in general reflects or might be in some part driven by this sort of populations being pushed south by glaciers, and then as glaciers retreated, populations of 6-line race runners generally pushed north, which reflects general knowledge of the herpetological movement in the US. And then zooming into our particular results on Indiana and Michigan, the first thing we notice is that our sole sample from Indiana groups in a clade with Michigan. So essentially this shows that the Indiana and Michigan samples are more closely related to each other than they are to anything else. And that group, or that clade, shares a most recent common ancestor with a sample from eastern Kansas. So as I mentioned, I wasn't able to collect any samples from Missouri, but Missouri shares a border with Kansas, and the sample that was closest to Missouri on the border of Kansas and Missouri, this 13277, is shown to be sister taxa to, or most closely related to, our populations from Missouri and Indiana. And we can see that this general group shares a recent common ancestor with this box in orange, which sort of represents the general region of Nebraska and Kansas and Colorado. But there's another way to visualize these results that isn't phylogenetic trees. And phylogenetic trees essentially show that Initial populations of six-line race runners occupied much of the southeastern United States, including Texas and Florida. And then we can see that in part related to the retreat of glaciers, populations began to expand northwards into states like Oklahoma and Arkansas. These populations continue to expand into states like Kansas and Nebraska and Colorado. Our results are also conclusive that these populations spread into Indiana, but our results sort of present two possible scenarios or support two possible origins from here. So either this current range naturally expanded into Michigan via a natural continuous range expansion, or populations were introduced or lizards were brought from this region that's sort of generally Kansas or Missouri, transported from here by humans recently into Michigan. So we can discuss a little bit more of these results. So without being able to directly sample from Missouri, it was difficult to test for the close relationship between Missouri and Michigan. So this figure above sort of displays the two possible settlement patterns that I've outlaid, and then what, based on our current sampling, what the topology or expected phylogenetic results would be. And the key thing to take away here is that without sampling this sort of ancestor of all the populations, we can't place it on a phylogenetic tree. So if populations in Missouri and Kansas are the ancestor of Indiana and Michigan, but we haven't sampled that ancestor, it's really hard to place it on a phylogenetic tree. So our expected result based on our sampling is this topology, which is the same for a native dispersal. So based on our sampling, our results support either an introduction from an unsampled population in the Midwest, like Missouri, or they support a natural settlement into Michigan. So the results do and are important for narrowing down the possibilities. So initially going in this project, we didn't know about the Missouri hypothesis. It was only through personal communications with collections managers that we learned about it. So we started off being like, they could be introduced from anywhere in the country. Now we've limited down to this Missouri region, and both my advisor and I have interest in eventually going back and collecting samples from Missouri and more definitively answering this question. So it really limits where and the scope of where we need to collect samples from. And importantly, improves the need or the call for more sampling, which probably have to be field work because these samples don't exist in natural history museums. And this type of problem is common when you work at range edges. Populations are being increasingly encroached upon by humans. And there are studies that show that these populations in Michigan or in Indiana are increasingly fragmented. So there's a need to sample these populations as soon as we can. And so because it represents half the lizard biodiversity in Michigan, and they're the reptiles, the smallest range in Michigan, and we didn't find any direct evidence for their introduction, we recommend that in Michigan, these lizards continue to be listed as endemic and protected under state law. I want to briefly acknowledge the funders and the contributors of these various organizations that really make everything happen. And a big thank you to my advisor, Dr. Bob Thomson. I would say half the time we spent together was just on developing my research interests and getting ready for grad school. And it was so much more than just trying to have me produce a usable product. It was really a mentorship about being an academic as much as it was about this project. And that's incredibly invaluable. And I want to thank him for that. And open it up for questions. I also want to mention that it's probably wondering why I ended up studying lizards from Michigan. And I think that a big part of it was their range actually covers New Mexico, which is where I'm from. So this is a picture that I took of this lizard because I thought it was cool in my yard before I ever started studying them. And I grew up like trying to catch them, and they're always so fast. And I was just kind of interested in the lizards. So when the opportunity came and I met with Bob and we talked about projects, I was like, how cool would it be to do my undergraduate thesis on a lizard that I caught as a kid in my yard? So that's how it all happened. Yeah, thanks. Open it up for questions.
Jeff?
What would have been done on tagging fragment?
Sorry, I did not hear you.
What work has been done on tagging track of individual items, regional tags, or something like that?
As in, like you're saying, to see if they're traveling. Yeah, that's a good question. In my personal literature search, I didn't see anything about tagging. I think I got pretty bogged down in trying to learn about genetics, and that ended up being a huge part of my literature review. so I can't answer that directly. I know that I don't think that anyone's ever noticed like them traveling large distances. I think they're generally considered to occupy pretty small regions, like in Tuscola County in that park. They're only found predominantly on like one singular hillside, and they're not really shown to like even move within the park. And they're not like a lizard that migrates or has seasonal migration. So I think in general, we can expect them to have pretty small ranges. But in terms of actual studies, I haven't come across any. Amber?
So you have this sampling problem. What about, is there any issues with the fact that you have these young, small, fragmented populations? Does that cause problems with trying to do the phylogenetic inference as well?
I think it does, and I think there were also limitations on sampling that trickle down into what type of analyses we can do. So we hope to do some other population level analyses. But yeah, I think I sort of forgot to mention in the introduction, but there are other sort of isolated populations of six-line race runners, including one in Indiana. And so I think when that can be sort of hard to parse out is initially we were planning to do sort of a genetic diversity piece, so sort of measure genetic diversity of lizards in Michigan compared to other places in the range. And I think that gets confusing when you have isolated populations. So those populations could be genetically not as diverse as the rest of the range just because they have a small population, even though they weren't recently introduced. So you start to see some of these sort of like confounding factors when you start to look at genetic diversity. But in terms of phylogenetic inference, I think that is probably a pretty robust way to test for evolutionary relationships despite sort of habitat fragmentation, because it's sort of using these like shared derived traits that you wouldn't find shared derived traits being sort of independently arose in other isolated populations. Like you would see them both sort of converging on having less genetic diversity, but you wouldn't see the same synapomorphies or the same sort of changes genetically.
I noticed that you were showing the dispersal of the lizards from south at the time that they seem to go kind of more central and then possibly veer back over more east. Does that have to do with like the Appalachian space?
Yeah, I'm not sure. That's a good question. I think a huge limitation to that was not really meant to be like this is exactly the dispersal patterns of six-line race runners. It's more of the samples that I have generally reflect this. I think that it could just purely be a result of people not collecting as many samples from the East Coast, because there have been reports of them as far north as Maryland in the East Coast. So, and like those samples weren't included. So like the sort of general path or dispersal path is as much driven on in terms of like there not really being an East Coast distribution is really, I think, largely just because samples from the East Coast haven't been deposited and as high of a frequency in natural history collections as much as it is the Appalachians or other factors. Roddy?
Yeah, so I'm just curious, all your samples, were they all like isolated populations? So in Michigan, they're only in this state part. Are they in other parts of Michigan and in other states where you got samples? Are they spread out in the whole state, or there's also in isolated samples?
Yeah, that's a good question. So within Michigan, there's only one population in the entire state. But from the rest of the range, it's sort of hard to know the answer to that question because many of these samples sort of cover a wide range of decades. Like I have samples from the 90s, and when those samples are collected, there's not like this metadata on like, this is how abundant they were, or this is how we found the population as well. So I think that habitat being fragmented is increasingly common in the last 20 to 30 years and is increasingly making these isolated populations. I don't have like definitive proof that any of my other samples are from isolated populations, but as I've shown that there's this other paper that came out in the early 2000s that's shown that the populations in Indiana are becoming more fragmented. So I think a cool future study would be sort of to sample some of those fragmented populations and do a comparison. I don't know if that answers the question.
That's a really good question, and unfortunately, it means that I don't want to. Use ERAD, which, you just, I'm sure struggle with, it's kind of a nightmare. And you generate it and beautifully explain your phylogenetics. But from this excellent question, it leaves how well can your phylogenetic data help you understand, like pretty low bootstrapping support within like the Michigan cluster. Are your other, and I'm really paying attention to this, but like how well do your other bootstrapping structures in the tree allow you to be answer that question?
Yeah, this is a good question, and it opens up a box of worms that I kind of tried to avoid. Like I intentionally avoided this because this is how you get on a rabbit hole, but it's an important thing to talk about. So yeah, so there's this whole other piece of the project in which I did something called a sensitivity analysis. So DD-RAD data, as you've mentioned, is incredibly hard to work with. It was very stressful for me trying to figure out how it even works conceptually, yet alone run it in a lab. A big problem with my data was that there was a lot of heterogeneity in sort of the read count per sample, in part because I didn't know how to use correctly DNA quantification thing. So I ended up loading like not the right concentrations of each sample into my library prep. So I had some samples that had like 20,000 reads, and I had some samples that had 13 million reads. So super heterogeneous. And one of the samples that was lowest on data was the sole sample that I had from Indiana. So that's that FMNH21. And so how that ties back into your question is that when we have a sample that has less data than other samples, it tends to sort of be less constrained in a phylogenetic tree. So that sample had the least of the samples included, the sample with the least sort of information or the least included sequences, the fewest loci in my assembly was that sample from Indiana. So in general, our expectations would be a phylogenetic tree would struggle to constrain it completely. So I think those low bootstrap supports sort of in the Indiana and Michigan clade are the result of less samples from Indiana compared to Michigan. So we can't place it within that tree because they're all pretty closely related. So each time it subsamples there, it's going to find a different result, but it was enough to sort of place it in that clade. And then how confident can we be sort of in the phylogenetic inference? I think we were worried about that same question. So we sort of we did assemblies and things in IPyRAD, and I did a sensitivity analysis, which is essentially lowering the amount of loci that are included or making your data matrix as complete as possible, really lowering the amount of missing data. And we found that the general topology of Michigan and Indiana being closely related was true and was supported by our phylogenetic inference based on smaller and less missing data sets. I don't know if that answers the question.
Other questions? Okay, thank you, Quinn. Oh wait, sorry, just on the line.
I was wondering, I guess, given how wide the range is from like Florida all the way up, like Texas to Mexico, up north, do they occupy a variety of different kind of environments and habitats for building different niches, or are they all kind of the same kind of forest habitat or maybe some difference?
Yeah, that's also a good question. I think in general, these things are not as well known about anything as they probably should be, particularly lizards. But I think in general, when the research showed they're often found in like not super forested regions. So like more open hillsides are generally where they're found. Like that's true from where I saw them growing up. So I think they're generally in more open, more grassland type systems. And that sort of ties in for how they could have transported from Indiana to Michigan. There's sort of evidence that there was continuous grassland in that region. So they are a habitat or a species that's more generally occupies grasslands more than forested regions. And there was plenty of grassland in that region.
Thank you very much, Quinn. Thank you for attending. We invite you to join us outside for food and drink and celebrate with our presenters.